Okay, so we're going to go ahead and get started. I have put up on the screen here a one-page handout that you have in your folder. This is not going to be kind of the classic idea of how do you put together a presentation. First, you have to have a first slide or an opening slide, then a background, then methods, then the results. I figure you already know that. On the other hand, what you do care about is how do you make the topic you're going to talk about extremely interesting to the people you're going to talk to. And we're in luck. We are trying to teach you how to talk to a group of people that will be judging your DataJam project. So you all have the exact same audience. And on top of that, you're going to be trying to teach students, the teams you're working with, the community college and the high school students, how to give presentations to that exact same audience, the judges for the DataJam project. So we can be pretty clear on who the audience is, and that allows us to really think about the strategies to get your message across effectively. I teach a university course about this every semester, and I was just calculating that I've been doing that for 15 years, so I've taught this class 30 times. What I've done for tonight is take a subset of slides, and I've drawn these slides from student presentations in the class. In this class, there's usually 40 students every semester, and each of them gives 4 talks, so that's 160 talks a semester times 30 semesters. I have an enormous number of slides to draw from. And so I've tried to draw some really good examples to teach you how to put together an interesting, effective scientific presentation following the principles that are on this spreadsheet. So let's start by thinking about what is on this spreadsheet. And perhaps what I'm going to do is just look at my computer here and point. So let me make this a little bit larger. Probably is more effective for the audience online than pointing to the slides in the classroom. So the very first thing you want to think about when you're trying to get people's attention and get them excited about your work is make really good use of your very first slide. Very few scientists do that. Scientists are actually some of the worst at getting people's attention. They might use a white slide with black writing, maybe a black slide with white writing, but we've all seen these, and they don't put pictures on it, they put their title. Their title often has fairly complex terms that cannot be understood until you actually hear the lecture. So if that's a title you're using, it's doing nothing to get the attention of the audience. And then has your name and where you're from. It's a huge waste of your first slide. What you really should be doing with your first slide is trying to focus the audience's attention on your topic and why your topic is so exciting. You can do that using a number of strategies. You want to have a title that's interesting to the listener before they know the details of your presentation. So something that really focuses on what you are going to be talking about using words that everybody can understand. Think about starting with a story or a question to engage the audience. Your brain is geared to pay attention to stories. And so many people giving scientific presentations say, I'm a good scientist. I'm not going to tell stories, but you should be telling a story because people pay way better attention to a story than they do to just factual information being handed to them. It doesn't work for every topic, but if you can make a story about why the topic is so interesting, and all of you in your proposals just had background information, that background information told a really interesting story. So think about on your first slide having a picture and telling the story that was in your background information of why you got so interested. People will be glued to your talk and they'll want to hear what you found out. It's a much more interesting way to start than just factually stating what your title is and going on. Questions are particularly interesting because you're a human being, and human beings' minds are designed to pay extra attention to questions. We know from measuring brain activity, either electrically or by imaging, that when you're asked a question, your brain immediately starts to try to answer the question. It is a fantastic way to get your audience to immediately start thinking about your topic. What I like to say is you start with an audience. They are kind of listening to you, but they're kind of not. They're scrolling through their phone messages. They may be talking to the guy that's next to them. They're thinking about what they need to do when they finish listening to you. And you're trying to get all these diverse minds that aren't really focused on your question to all be thinking exactly about your question. So using a question, then they can't help it. Their brain starts to answer the question and you've immediately got them all thinking about the topic you're going to talk about. I strongly suggest using a picture on your first slide to help animate what you're going to talk about. Much more effective than just having words on your first slide. Under some circumstances, you might want to have co-authors. I don't think we're going to talk about that today. You're going to have your team. And the last thing that's listed there is something that scientists have a lot of pushback against. They generally think, I'm a good scientist. I am not going to evoke emotion. But again, people's brains are designed to not only pay attention to questions, but to pay attention to emotion. And if you can get people to kind of go, oh, that sounds serious, or oh, that's too bad, or that's unfair, or that's awful, or that's funny. It doesn't matter what the emotion is. Evoking emotion will get extra attention. And so you don't want to shy against that. You want to be professional and you want to be a good scientist. But if it's possible to build in evoking emotion, that works great. And I'm going to show you a number of examples, again, taken from slides that all of these thousands of students have produced over the years I've been teaching on a wide variety of topics. So hopefully we'll have something that everybody can relate to. You not only want to engage attention with your opening slide, you want to keep engaging attention throughout your entire talk. So you can ask questions, but usually audiences are not thinking they're going to answer a question in the middle of somebody's talk, but you can ask a rhetorical question that does the exact same thing. What you have to do when you ask a rhetorical question is ask the question and then just leave a momentary pause So everybody's brain starts trying to answer the question. You've got their attention, and then you go on and you answer the question. If you can involve the audience somehow, if you can get them to look at something or hear something or do something, that is a very good way to get the audience's attention. You want to be animated. If you just stand there completely still and you have very little facial expression, you don't use hand motions, then you will be fairly boring to listen to and look at. So you want to use facial expressions, body language, hand motions. You want to be interesting to look at. And again, if you can, evoke emotion. Very frequently, people get very set on talking about methods. And I've seen this happen in DataJam presentations time and time again. The DataJam teams are so excited about all the analytical methodology that they've learned that they spend an inordinate amount of their 10-minute time talking about methods. And that is very boring. You want to talk about the methods you used or outline them, and we'll give you a number of strategies for how to do that effectively. But you then want to move on to the results and talk about how those methods allowed you to find the results. The focus of the beginning of your project should be on why, telling a story, or why did you do what you did? Why was it important? Then a little bit about what you did and how you did it, with a big focus on what were your results, and then on what you think they meant. So the three big areas to pay attention to in your 10-minute talk are: why was it important to do what you did? What did you find? And what do you think it means? That will make a story that people are very interested in listening to. So we say you want to use just enough methods that people can understand the results. And I'll show you a bunch of examples where using a timeline or a schematic diagram or something that they can look at that isn't words to describe your methodology is really, really valuable. You want to get your message across effectively as you show your results. So a very good way to do this is to put your graph or your diagram that shows your results on the slide in a way that's easy to understand and is very well labeled. But put the take-home message, what you think it means, at the top or the bottom of the slide, so people can see what you did and what you got, but they also gets reinforced by a statement of what you think it means. At the end of your talk, you really want to emphasize your take-home message, and you want to think about, how am I going to get this across visually? How am I going to get it across for auditory learners, people who want to hear the message? And how can I get it across for people who want to envision things and are more manipulative? So you'll notice lots of scientists use bullet points at the end of presentations. I'm not a big fan of bullet points at the end of presentations. I think you can get your take-home message across much better most of the time with a schematic diagram. And I'll show you a number of examples of this kind of thing. If you can link the end of your talk to the beginning of your talk, and you can make a complete story, people will remember your talk much better because they heard the whole story. They may forget a couple of the details, but they'll get the general gist of it. So thinking about linking the end to the beginning is a good idea. Okay, so you all have this. I like to have this right beside me when I'm making slides for my talk, so I try to remember to do these things. I'm going to pull up the series of slides that you have, and you have these. You can look at them. I'm Judy Cameron. I am a neuroscientist and I study the brain. To get across to you who I am, I've put a picture of the brain here. It's moving, it's colorful, it looks a little weird. This is a technique that was actually developed at the University of Pittsburgh originally, but it's now used all over the world, looking at white matter tracks in the brain. So this is diffusion tensor imaging that allows you to see all of the connections between brain areas. And what I'm going to talk about now is how do you get your message across? Because that's exactly what you care about. That's what you want to know. Pick a title that dramatically says exactly what you think your most cool take-home message is and what your audience wants to hear about. So the first few slides are going to be just examples I pulled from different people's talks. How do you engage your audience with your very first slide? And because I'm giving this, I chose a slide from one of my talks. I do work on the effects of exercise on protection of the brain, neuroprotection, and its ability to prevent neurodegenerative diseases with aging. And so this is a talk where I was talking about the fact that exercise is neuroprotective. And I titled it something that everybody could understand. Exercise is neuroprotective. This was at an international meeting at the Parkinson's Disease Foundation, and I was the only speaker talking about doing research in non-human primates. And so the second part of my title says, New Findings in a Primate Model of Parkinson's Disease. And because almost nobody did work with non-human primates and nobody has an idea of what non-human primates would look like when they're running, I have a little video that shows non-human primates running on treadmills. Usually this gets a little bit of a laugh from an audience that has never thought about this before, and that's evoking humor. You're being a serious scientist. You're not overtly evoking humor, but you are almost always getting smiles for that. If you look at the color of the slide, it is subtle, it's not bright, but it's also not black and white. We emphasize the title using yellow. The background is green. It was designed to match the scrubs this person was wearing. But if it looks a little bit different and it's interesting to look at, people will be paying a lot more attention. It might only take you 10 or 15 seconds to say what you want to say with this title slide, but people will be glued to it. They will be focused on your topic. So think about using pictures. Think about using animations. Think about color. Think about a title that people can relate to. Think about evoking emotion. Here's a slide that a student in my class used as an opening slide, and this topic was treating LEMS. And what she said was, you probably all wonder, what is LEMS? And of course, nobody in the room knew what LEMS was, and everybody was wondering what is LEMS. And the fact that she said it with expression and she asked it as a question, you know that every single person listening was like, Okay, what's LUMS? They're all thinking about your topic. It is a fantastic way to get them all to focus on what you want them to focus on immediately. She said, look over here at this man. His knee seems to be hurting. Maybe it has something to do with joint pain. Well, in fact, it did have to do with joint pain, and it is a disease process where there is a breakdown in neuromuscular communication. And she went on and gave a talk about what they were doing to understand the etiology of it. But fantastic slide to get people's attention. Also, he's kind of grimacing, and you're like, ooh, it probably is painful. And you kind of feel that emotion. Again, not being afraid to evoke emotion to get people's attention. There's a green and blue bar at the top, just adds a little color, makes it different, gets attention. Here's another student who was going to talk about Alzheimer's disease, and he used a series of questions. Today we're going to talk about what is it, who will develop it. So a series of questions, getting the audience focused on that. He talked about a movie that had come out where this woman has Alzheimer's disease. Her husband very nicely comes and reads to her every day, but she doesn't remember anything, so he reads the same page over and over. And when you hear that story, it's sad. So again, you're evoking emotion, but not as much emotion as when he talked about this picture. After a while, you are so isolated, it's like you're walking down the path of life all by yourself. You can't remember your friends. And everybody in the room, of course, was like, oh my gosh. So you very quickly get people's attention. And that's what you want to do with your first slide. Here is a slide about I told you about diffusion tensor imaging in the first picture I used. This was a talk by a student talking about this, and they played this video while they were talking, and you're going to see him fall, and you could tell exactly when they fell because the whole audience kind of lurched back. Again, evoking emotion to get attention. Here's another example. This is looking at sleep depth and fatigue, the role of inflammation. This little girl has inflammation. She's having fun playing in her playroom, but she's so tired that she lays her head down on the shelf and falls asleep. You're using storytelling combined with emotion to get people to really care about the topic you're going to talk about. So you want to think about your own DataJam project. You all just talked about your DataJam projects. How could you introduce your DataJam project in a short 30-second introduction, but that has a picture, has a story, has a question, evokes emotion to get the audience, the judges, to really care, to pay attention to your topic. Now, you want to present the background in an interesting way, and the worst possible way to do this is a list of bullet points. Since I've been teaching this course on giving presentations, I go to tons of scientific meetings and I look to see what do people do when somebody puts up a slide with bullet points. And I can tell you more than 75% of the room picks up their phone and starts scrolling through their phone. They instantly stop paying attention. So the worst thing you can do is a list of bullet points. The most common thing that scientists do is put up a list of bullet points. So you don't want to do that. There are a lot of other strategies you can use. Again, I'm going to give you examples from my classroom. This is somebody talking about cognitive impairments in multiple sclerosis. That's what MS is. They do have bullet points, but they're really, really short, so they're not awesome to have to read, and it's not boring. They have a picture of a woman, and she has a neuron next to her in the brain. And they told a little story. They said the worst thing about multiple sclerosis is that it leads to cognitive problems that affect daily functioning of the patients in many ways, weakening their ability to work, engaging social functioning, quality of life, and increasing the strain on the caregiver. We're going to tell you about a specific population of neurons in the brain of these people who really are suffering. So it's given you a capsule background of why it's important, has evoked emotion, has given you the main symptomatology, all in one slide that is not boring to look at. Timelines, or things that you can follow, are really good ways of giving both background and methods. So this is a talk about a mechanism of a motor decision. How do you make the decision to throw a ball or move your arm? That's what a motor function is. The message starts in the premotor cortex. It sends the signal to the primary motor cortex, to the cortical spinal tract, and it synapses on your skeletal muscles that actually make the movement. And this talk's going to focus on the premotor cortex, and there's a picture that shows where it is. Again, combining background information with a picture to really get people's attention. You can also use animation in slides. So this is looking at glucocorticoids. GCs are glucocorticoids, things like corticosterone, cortisol. They act through receptors through two pathways in cells. The first pathway is to bind to the DNA in the cell. The other pathway, so that's called genomic signaling, the other pathway is to bind to the cell surface of the cell and activate second messengers, and those will have an impact. So kind of showing step by step, and I'll show you a few more examples of this, but using animation can be very effective. Like I said, you want to present just enough methods to understand the results. And so I'm a huge fan of using timelines. And here, what you're doing in this study, they collected brain tissue and they wanted to look at certain cells in the brain tissue. So they collected the tissue here, they dissociated it into cells, they treated the cells with growth factors and made what are called neurospheres. They dissociated those and treated them again and then plated them into wells. They do have bullet points, but they're just 3 bullet points, and it's telling you that they treated these cells with a concoction of things. They gave ethanol as a control, and then they harvested the cells at 1, 3, and 5 days after drug treatment. It's interesting to look at. It's easy to follow because it's a timeline and it has pictures, and you get a very good sense of what they did. Here is a talk about fruit flies, and you would think looking at behavioral studies in fruit flies would be really boring. But I think they made the methods really interesting. They expose them to mechanical changes, and they look at who's sensitive to mechanical sensitivity. And they just put in an animation of shaking the tube. People laughed. So you've got their attention. They'll not forget what you did. And they're amused, so they're paying extra attention. or temperature sensitivity. And they put the tube in a bath with a rubber ducky. Now, of course, in the lab, they actually put it in a very boring looking water bath. But showing it this way, again, got a smile, and you're not going to forget how they did this. Here's another example of a timeline, taking a tissue section, collecting the cells with a new technology, laser capture microscopy, and they put a blow up over here of what that is. So this is a part of the brain they were studying. They stained and identified the cells they were interested in. They used a laser to cut them out. So here you see the cells are removed, and then they've put them over here. So now you have a really homogeneous population of these cells. And they then extracted the mRNA and did microarray profiling. So they're combining a timeline with an inset picture where you actually get to see exactly what they did. Here's another animation. You're looking at a timeline that's animated. This was taking elderly patients diagnosed with depression. They're treating them with an antidepressant. The problem with antidepressants is it can take many, many days or weeks for an antidepressant to alleviate depression. And everybody would like to know, is there an early marker of who's going to respond to an antidepressant and have less depression and who isn't going to respond? So they took a series of brain images, MRIs, and then they divided them into treatment responders and treatment non-responders, and they asked, can we see some early indicator of who fell into which group with these MRIs? So they've used animation to walk you through a timeline. You know exactly what they did, and you know it very, very quickly. Here is another animation. This is neuroimaging again. They have had a person exposed to a story where they have to show emotion regulation, and they've identified a part of the brain, shown in red here, from a number of different angles that is activated with emotion. And then they use a schematic diagram to show you exactly where that spot is. So this is kind of the background information and the methods combined, and the whole rest of the talk, you're going to look at what is going on in this part of the brain. Okay, we've made it through the whole front part of your talk, and hopefully you have ideas that you can translate to your own DataJam project. I realize we're going fast and we're just giving you single examples, but you'll have these slides that you can look at, and you can think about, how can I be more clever about putting my slides together so they're interesting? You want to present your results clearly. Again, this is a slide that I made, that I use. This is exercising monkeys to see what effects exercise has on the brain. What we did is we had monkeys run for 5 months. They got injected once a week with a compound called BRDU that gets incorporated into newly made cells. And so it's stained green. This is a slice through a brain tissue. This new neuron is green inside, so it was made while the animal was running. We looked at a whole group of middle-aged monkeys and older monkeys, and running increases production of neurons in the hippocampus of middle-aged animals. But you have to run while you're middle-aged, because running later in life doesn't do the same thing. And the take-home message is right there at the top, so people can see what you did, what results you got, and what your conclusions are, all in one slide. This was a study that a student presented. They're looking at lifespan, again, of Drosophila, and that have different mutations. And you can immediately see that they go from all being alive to all being dead down here, and that these genetic mutations really change the lifespan. So putting all the comparisons you want to make on one graph or one slide is a really good way to help people look and see how things compare. You could put them on different slides, but then they wouldn't be able to compare them directly. Here's another example of doing the same thing. This is looking at brain tissue histology in a number of different animals. And right off the bat, because you have them to compare one to the next, you can see that they look really different. And that tells you that the genetic mutations really were having a very big impact. I think that tables are the worst possible way to present data. It is boring, and people, again, pay almost no attention. If you're going to use a table, you probably want to highlight what you want people to look at. But again, it will be your least effective way of getting a message across. This is another study, and this one is frightening. But the fact that the images are all next to each other make it really easy to look at. This is a person who had a TBI, a traumatic brain injury. So this is the right and left side of their brain beforehand. This is 4 months after an injury, and you can see they lost all the white matter right here on this part of the brain. And the thing with white matter is you don't get it back. It does not grow back. You may get brain function back, but it's because other parts of your brain take over the task. You don't heal neurons. So here's 10 months later, they're still completely missing. Very impactful, because you can have the pictures side by side and people can look at it. And in fact, when you present this, people feel an emotion. They're not going to forget that. You probably won't forget it, and probably none of you studies traumatic brain injury, but it's bad. You might think, there's some things you just can't make interesting. This is a chemist who gave a talk, and I thought, this will be hard to make interesting. But they were looking at neural activity with different compounds. They color-coded the compounds. They circled what the differences were between these compounds, and then they showed that they have really different physiological effects. So because it was color-coded, you could look right at what the molecule was and see what the physiological effect was. Showing things like tables is really hard for people to grasp, but things like blots is even harder for people to look at. They look at all these blots of proteins and they're like, I don't know, this doesn't look different. But if you graph it, it looks much different. And if you graph it and you use a red circle to draw people's attention, then people will get the message. So try to use strategies where people can visualize what you're trying to show. And at the very end, you want to emphasize your take-home message. So this was the last slide in a talk where the very first slide was a bunch of soldiers that looked like they got shot up coming off the field. in a military operation. And what they said was, all of these people have traumatic brain injury, and we need a compound, something we can inject right on the field that will minimize brain damage and can start to treat them immediately, because it takes hours to transport them to field hospitals. Their whole talk talked about this compound, N-acetylcysteine, but I thought they did a phenomenal job with their conclusion. They wrote out a sentence and they said early treatment with N-acetylcysteine dramatically improves patient recovery. And then the clever thing was they had a picture of this soldier walking off the battlefield looking totally intact and in good shape. So they were able to link the end of their talk to the beginning of their talk. And you could contrast these dilapidated people coming off the field with, if you treat with this, we have a much better chance of getting them cured. Very, very powerful end slide. Here's another example of a concluding slide. No words at all. This is that study where you were looking at brain trauma at 4 months and again at 10 months, showing you didn't regrow brain pathways. And the conclusion was, we need to use this imaging technique, diffusion tensor imaging, because it tells us where the damage is and how bad it is. And just the picture itself told the message. Very, very nice. Here was the ending slide from a talk that talked about the fact that you want to use implicit memory versus explicit memory. Implicit memory, they started their talk by showing a picture of Peanuts cartoon of a kiss and said, how many remember their first kiss? And I thought, this is very inappropriate for a scientific talk. But I listened, and everybody raised their hand. Everybody remembered their first kiss. How many of you remember what you had for lunch yesterday? A few people raised their hand. How many of you remember your credit card number? Nobody raised their hand. And the idea was, if you take people that have had memory problems or traumatic brain injury and start to get them to remember implicit memories, emotional memories, it's much more effective. They finished their talk with this slide. Our new rehabilitation strategy is to focus on the implicit advantage, emotional memories. And they had some pictures that just got you to understand what that was. So better than bullet points, some visualization. And all three of these ending slides basically have been visualization strategies to get your message across. So that's your quickie lecture on how to make your slide presentation much, much more interesting than it would be. I think it would be wonderful if each of your teams tries to put some of these ideas into action. The more you do, the more fun it will be for your audience to listen to you. And if they're judges at a DataJam competition, the judges will think much more highly of your work. Yes, they'll pay attention to what were the analyses you did, and did you make good conclusions from your analytical results. But if it's an interesting story and they get really excited about it, they're much more likely to have a good opinion of what you've done. So you want to be able to not only put this into action with your presentation, but to be able to teach the teams you're working with how to do that. Okay. Any questions? It's a lot to digest all at once, but I wanted to give it to you now because I think you're going to make good use of it for your second DataJam project. See you next week. Okay. Good night.
Thank you.
Thank you. There you go. A whole course in 1 hour.
I'm trying to